For everyone in the U.S., happy Juneteenth Eve, and welcome to the Temporal Genomics seminar. Thank you everyone for coming to this Friday seminar. My name is Brendan Reed. I'm a postdoc in Malin Pinsky's lab at Rutgers University, speaking to you from the traditional territory of the Lenni Lenape people. This series is organized by the Research Coordination Network for Evolution in Changing Seas Temporal Genomics Working Group. If you want to learn more about our working group, we have a lot of information on our website. I'm going to link that in the chat shortly. But thanks for sticking with us through the series. This is our penultimate seminar. Both of these final seminars are going to be roundtables where we have 4 different researchers presenting their research. The final seminar is going to be back on the normal day, Thursday on July 1st at the same time. And we're also still planning to feature additional research the following week. So we'd originally planned a Twitter conference. It will probably take the form of a single day where people are tweeting out their research. If you signed up for that, we will be letting you know more about that shortly. If you haven't signed up, we're definitely still interested and we have space for additional presenters. So feel free to get in touch with one of us if you're still interested. Just a few ground rules. While our speakers are presenting today, we ask you to please be respectful. Please keep yourself muted and hold all questions until after the presentation is over. And once our speakers are done, we ask you to either use the raise hand button or indicate in the chat that you would like to ask a question. We're going to hold all the talks till after, or all the questions till after everyone has spoken. So at the end of the seminar. Katrina Catalano will be helping me moderate, so either she or I can call on you, and then Joel or Yoel who's also who's also doing the work of moderating, could call on you too. Yeah, if something unforeseen happens, we have an alternate webinar link, so we'll do our best to email that link out to you quickly, and then resume as soon as possible in the webinar. So okay, cool. I think with all of that out of the way, let's welcome this week's speakers. I'm going to introduce our first speaker, and then they can take it from there. So please welcome Dr. Joel Alves. Joel received his bachelor's in science and a master's in biodiversity from the University of Porto. He completed a PhD in genetics at Cambridge and went on to do a postdoc there, as well in conjunction with CIBIO in Bio in Portugal. And currently he's a postdoctoral researcher in the Department of Archaeology at Oxford. So let's welcome Joel. And you're muted, Joel.
I'm going to start all over again. So first of all, thanks, Brendan, for the very kind invitation to be here hosting. I won't present anything because we had 1 hour to present to our presentations, and I thought it would be much more interesting to hear from these 3 different people. So it was quite a sneaky way for me to listen about people that I'm quite interested in their research. So the objective of the seminar was basically to see how we can detect selection with time. The ability to generate a lots amount of data, and especially for temporal data, is increasing with time. And so there's novel methods and there's new questions that are arising. And so I also like to thank the 3 speakers that we invited to be here, Alison, Evan, and Bastiaan, to kindly agree. And what's interesting about this sort of format is that we'll start, it's not only 3 speakers, it's 3 different models, and there's sort of 3 different time scales of evolution. We'll start with virus, moving to cod, and sort of ending in humans with a much sort of larger scale. And as Brendan said, I'll introduce 1 speaker after the other, and then in the end, there'll be time for questions. Feel free to write them or just unmute yourself and talk, but you also feel free to write them on the chat while it's going, and we'll read them in the end. So we'll start with the first speaker, which is Alison, that is now an assistant professor in the Department of Genome Science in the University of Washington. She just started this position, and she has really fascinating research on the topic of HIV evolution. So off you go, Alison. Thank you once again to agreeing to be here.
Yeah, absolutely. And everyone can hear me and you can't see yourselves? Great. Yikes, 1 second. Just go to presenter view. Awesome, cool. Yeah, thank you so much for the introduction and the invitation. I'm excited to be here on the shortest end of the time scale. So as Joel said, I'm a new faculty member at the Department of Genome Sciences at the University of Washington, and I'm really interested in understanding the dynamics of rapid adaptation. And one of the reasons that I find it so fascinating to study rapid adaptation is actually simply because you can watch evolution happening in real time and measure it with things like time series. And there are a few systems where this rapid evolution has been so devastating than in the case of human immunodeficiency virus, or HIV, where we have in real time produced drugs that can combat the virus only to have the virus evolve around them in a matter of weeks. And I want to jump right in by showing you more concretely just how fast HIV can evolve. So we're going to consider a population of viruses inside of a patient who has never seen any drugs previously, who was treated with an early HIV therapy called 3TC. So drugs are applied continuously starting at week 0, and this patient is followed over time. And we can assay the virus or assay the patient for large effect drug resistance mutations in the blood and watch how the population composition changes over time. And very quickly, what we see is that a drug resistance mutation goes from being totally undetectable in the population to being fixed in the population. So it's clear that in this patient infected with HIV, time series data can show us that evolution can be strikingly fast. Evolution is not only fast, however, but also predictable. So here are 18 additional patients treated with the same drug and plotted in parallel. And what we see is that in 80% of these populations, we see drug resistance above 50% frequency in 30 generations or fewer, with potentially even more individuals having drug resistance happening in this time scale where we don't have time points at the correct measurements to actually say this. So these data make it abundantly clear that evolution can be really fast and really predictable. And the questions that I want to dive into today are, what can these extremely rapid trajectories tell us about what's going on sort of under the hood in these populations? So I'm going to tell you really two brief vignettes. First, how can time series data help us predict genomic signatures even with extremely minimal sequencing? So we don't really have genomic data in the time series that I just showed you. We sort of have single drug resistance mutations. But I want to tell you about how this can still be quite useful in terms of bounding population parameters and kind of giving us a sense of what's happening under the hood. And second, I'm going to move to a more traditional example where we use sort of standard genomic data in order to estimate HIV population parameters. But towards this first question, what we would like to do is quantitatively understand what's going on within patients for drug resistance to rise within 30 generations so predictably across patients. And to do this, we start with just the simplest Wright-Fisher model with selection and ask, what population parameters can produce these trajectories. So in this model, we imagine we have n viruses, and each virus has a probability mu of acquiring a drug resistance mutation within each generation. The product of these two terms, n times mu or theta, tells us the number of beneficial mutations we might expect to be entering the population each generation. And then we imagine that these drug resistance mutations provide a selective benefit 1 plus S relative to the wild type. Then we can just sort of simulate under the simple model and ask, under what sort of parameter combinations are we able to see sweeps that happen so quickly and so predictably in 30 generations? Remember, what we saw previously is that sort of in 80% of populations, we see a sweep in 30 generations. And so when we actually do these simulations, we see that this time series quite constrains the area of the population parameter space into values that are actually quite sillily, like funnily large. So the selection strains have to be, enormous in order for selection to happen so quickly. But in order for selection to happen so predictably, we actually also have to see that the population mutation rate is quite large as well. So the timing and consistency of sweeps suggests that we kind of have to be somewhere up here. And the next question I want to ask is sort of, what does a sweep look like if it's happening so quickly? And in our conventional model of adaptation, we imagine that we have some sort of a change in environment, like a drug being applied, and the population has to wait for some period of time before a beneficial mutation is created. And this happens sort of with the inverse of the population mutation rate. And then a beneficial mutation appears and it sweeps through the population in your classical hard selective sweep signature. Now, we're thinking about populations where we think that theta becomes quite large. And if this is the case, then we might have a much shorter waiting time for a beneficial mutation. And so as this mutation appears and begins to sweep in the population, this waiting time for another beneficial mutation might be shorter than the amount of time it takes for this beneficial mutation to sweep. And if this is the case, we might have a secondary beneficial mutation entering the population and beginning to sweep in parallel. So this pattern, in which multiple beneficial mutations sweep simultaneously, is known as a soft selective sweep. And it turns out that understanding something about really rapid and predictable evolution can actually also tell us something about how likely we think soft sweeps should be happening in our data. So here again is this plot I showed you before, showing the area where we're likely to get a predictable sweep in 30 generations. And we can ask for each of these successful sweeps in 30 generations, how likely is it that those sweeps were soft? And what we see is that the area where it's likely that we get a sweep in 30 generations is sort of overlapping with this area where it's likely that those sweeps should be soft. So this is telling us that in order for evolution to be so rapid and so predictable based on these time series, that it's actually likely that multiple beneficial mutations have been produced and are spreading simultaneously. So this is a prediction that we can make about our data, even from these really high-level trajectories where we don't have any information about sort of what's going on at the genomic level. And I'll show you an example in a second about how we can actually look into the genomics themselves in order to actually try to assess whether these soft sweeps are a prevalent feature of HIV. So that's sort of the short answer to this very short vignette about how these really high-level time series can help us. I want to move into a more traditional example about time series genomic data and how we can use it to estimate HIV population parameters. So as before, we're going to think about population size and the strength of selection, but we're going to add in this additional dimension, which is the rate of migration. So HIV inside of a patient is not just some sort of well-mixed population, but is a series of subpopulations in different anatomical locations interconnected by migration. So one of the things we're going to try to do is actually use time series data in order to estimate that rate of migration in a way that would be really challenging to do if we only had sort of single time point equilibrium data. So this is, I'm going to be showing you data that was produced by Zandria Ambrose at the University of Pittsburgh and her team. And she did a macaque study in which she infected macaques with RT-Shiv, which is a simian immunodeficiency virus with an HIV reverse transcriptase spliced in. So this means that the virus is able to replicate pretty normally in these macaques, but you can treat it with HIV drugs and it gets the same types of resistance mutations as we would get in humans. So this macaque was initially infected and the virus was allowed to establish in the macaque, and then the virus was treated with a monotherapy, so sort of similar to the types of drugs I showed you in the previous example, which initially succeeded but ultimately failed in suppressing the viral population. And what Zandria did is she sampled it at multiple different time points before, during, and after the evolution of resistance. But what I think is actually really exciting about this study is that she also sampled for multiple different anatomical locations, including the plasma, the lymph node, and the gut, which are all what we think are important reservoirs for HIV. And at each of these different time points and spatial locations, we have approximately 30 single genome sequences that we can use to try to understand the dynamics going on inside this macaque. And I'm going to just jump straight into showing you the data. So this is sort of a Muller-like diagram coming from the blood plasma. And so we have time on the x-axis, and we have the frequency of these different viral genetic variants on the y-axis. And what we see is that initially at the first assay time point, the population is initially entirely wild type. But then before very long, it's same in time scale of 30 or so generations, we get the fixation of multiple different drug-resistant haplotypes. So here, and all of these underlying variants are known assayed drug resistance mutations in HIV for this particular drug that was given. So we see that, as we may have predicted, the sweep appears to be soft. We have at least 2 drug resistance mutations, M184V and M184I, spreading simultaneously. And perhaps this is also another independent origin where we can't tell these two things apart. Maybe this synonymous mutation happened on the background of M184V, maybe it happened before. So what this is suggesting is that, sort of consistent with the timescales that I showed you previously, sweeps do seem to be soft and selection does seem to be extremely strong. But one thing that's really exciting about this data is that we can also look at the same different dynamics going on in the lymph node and the gut. And what we see is that the same types of viral haplotypes that we see spreading in the plasma are also spreading in the lymph node and the gut. So most of the major genetic variants that we see in one location are found in all of the other locations. However, suggesting that the migration rate between these different compartments needs to be relatively fast. On the other hand, we see that the actual trajectories within these different subcompartments are not doing exactly the same thing. And that, in some cases, this M184V variant is increasing, and in some cases it's shrinking. So qualitatively, we see that mutation selection and migration are all apparently strong relative to each other. And the question is, how can we sort of make this a more quantitative observation and actually try to put some numbers on these parameters that can help us try to explain why we would see, for example, haplotypes that are shared across the compartments but are doing different things. So to do this, we're going to go back to our favorite simple Wright-Fisher model with selection, but we're going to add in just the slight wrinkle that we're going to be thinking about a series of interconnected populations where they have sort of the same dynamics playing out. So the only parameter that we've added here is n, this migration rate, which tells us the probability for each virus here of it migrating into the other compartment. And then once we have these, this model, we can simulate it under this model again and try to ask under what sort of under what sort of parameter combinations are we able to produce similar types of trajectories that have these same types of features we observed in the SHIV data. And so we're going to use approximate Bayesian computation in order to estimate these genetic parameters. And so the high level picture of what's going on here is that we have prior distributions over theta, S, and M, which is to say the population mutation rate, the selection strength, and the migration rate. We can sample a particular set of parameters from these different prior distributions, and then use it to simulate time series data that looks or may not look kind of like the SHIV data that we saw previously. We can sample them in time series and summarize these data in a variety of ways. For example, computing things like the FST at a bunch of different time points, or computing the number of shared haplotypes above a certain frequency. And then we can compare this to the data that we found in the SHIV scenario and try to see whether it's similar or not. From that comparison, we can choose to either reject or accept this particular set of parameters that have generated the simulation. Now, if we accept it, we add these particular parameters to the posteriors, and if we reject it, we just sort of start all over again. And we repeat this process until we have derived posterior distributions that can help us understand what are the types of input parameters, theta, S, and M, that can produce data that's similar to what we have in the SHIV's case. And what I'm going to show you are these posterior distributions. And I'm going to do it pairwise, such that we're comparing the plasma to the gut and the plasma to the lymph node. And the reason that we do it pairwise is because we've done a bit of theory to try to understand the dynamics, and we've only done this in a pairwise fashion, but there's no reason in principle this couldn't be extended to three-way comparisons. So for the selection strength, we once again see estimates of S that are between 1 and 10, again, hugely enormous parameters that actually may make sense in the context of HIV. And we get a similar estimate when we do the same comparison independently on the plasma versus lymph node. For the population mutation rate, we get estimates that are sort of between 0.1 and 1. And these are actually quite consistent if you take these estimates to what we would have estimated in the first half of the talk, saying that these are parameter combinations where we're likely to see a sweep in 30 generations, and that sweep is reasonably probable to be soft. But where it gets a little bit more interesting is actually when we go to estimate the migration probability. So here is the migration probability between plasma and gut, and here is between plasma and lymph node. And what we're seeing is there's actually a separation where it looks like there's more migration that connects the plasma to the lymph node than the plasma to the gut. And this is a potentially interesting observation because we think that one really important factor of how HIV may be evolving multiple drug resistance, which I haven't talked about at all today, is that there's sort of dynamics that are complex within patients about how drugs are reaching different places, and understanding how fast viruses are moving between these places could be an important component. And one thing that's really sort of interesting here is that in this long-term neutral equilibrium of where we would expect to be able to estimate migration rates, above this NEM equals 1 point in the, we would expect that it would be quite challenging to estimate migration rates. But here what we're seeing is that because we're actually looking at dynamic time series data, we're measuring forces of migration on the time scale of selective sweeps. And because we're actually able to sample them in multiple times, we're able to sort of reach into this area above NEM equals 1, where we think it should be really difficult to estimate migration in the sort of neutral equilibrium. Okay, so I should mention that we are, I'm going to finish up in a second, but if these sort of questions sound interesting to you, I want to say that we have a lot of ongoing work, and this, as Joel mentioned, we just opened the lab at the University of Washington, so please be in touch if these questions sound interesting to you. I want to just sum up really quickly, saying that we can see very predictable evolution in HIV, which suggests that HIV drug resistance occurs at least partially via soft selective sweeps, and that we can estimate intrapatient population parameters via approximate Bayesian computation in order to quantify and distinguish really fast migration rates that we hope are going to allow us to understand something better about how multi-drug resistance evolves. And with that, I want to thank my collaborators on these projects and the organizers of the symposium and Joel for allowing me to speak today. And I'm happy to take any questions, I guess, after the other two talks or in the chat. Thanks very much.
Thank you so much, Alison. This is a great presentation. So we'll move to the next speaker, which is Bastiaan Star, that is now an associate professor at the Center of Ecological and Evolutionary Synthesis in Oslo, and will talk to us about COD and the detection or non-detection of selection in the populations. Off you go, Bastiaan.
There you go. All right. I'll just share my screen, guys. Can everybody see this? Oh, it stopped sharing. Or did it not share screen again? There we go.
Yeah.
Does this work all right? Sweet, Brendan. So yes, I'm really happy to talk about the temporal genomics that we do here on Atlantic Cod. I think you have a really wonderful seminar series. I've been seeing some of the previous speakers, and it's really an honor that I can try to contribute to your environment here. So I work in the University of Oslo, and the temporal genomics I'm going to talk about really comes from an interest that I had for a long time about the long-term exploitation of fish in the Northern Hemisphere. And although I focus here on the Northern Hemisphere, I think this is a picture that we can see in many different parts of the world, where we see that we have in the Stone Age subsistence hunting on fish, which then sort of developed slowly into early trade, especially for us here in Norway during the Viking Age. But then during the Middle Ages, at least in Europe, we really see a fish horizon appear, where we find lots and lots of different bones in different archaeological settings, and whereby larger and populations further away are being increasingly targeted and transported. And this really gave a tremendous amount of wealth to several nations here in Europe. And of course, this then has further escalated in the global industry that fishing is now. With a complete globalized industry, stuff gets moved over the entire planet. We have a massive capacity for fishing right now. And that really happened during sort of the last 20th century. And this is what I'm going to focus on in this talk. So what happened during the 20th and the early 21st century is that we have a lot of capacity. There are a lot of populations that are being completely overfished. But apart from that, we also see major phenotypic changes in a large proportion of heavily exploited fish populations. I'm just wondering, do you see a black screen on the right of my shared screen or not? Do you just see the screen? Okay, I'm just seeing your images over part of my presentation, that's why. So what you see here on the right-hand side of the screen is the phenotypic changes in age of maturation that we find in Atlantic cod. And this is a very important sort of character for fish, because the moment you mature, you start reproducing and you stop your energy in the reproduction, which means you don't grow as large over your lifetime. Some of these changes phenotypic changes are some of the largest we can record in natural ecosystems, or semi-natural ecosystems, I may say. And this, of course, has been of concern. And the concern has been whether these changes have been driven by evolution and by fishing for these populations. So this is not a new idea, actually, and it was already J.S.B. Haldane who thought that fishing could be such a force, such an evolutionary force that could lead to extreme and abnormally fast evolution that we might observe in our lifetime. This is, of course, what we now call fisheries-induced evolution, even though he did not use the term himself. So what evidence is there for fisheries-induced evolution? Well, of course, we have a lot of theory, and there is a lot of mathematical models that describe the impacts that we may have in size of fishing. And of course, it reads like a Darwinian cookbook. You fish, you have populations, you have fish of different size. Often fishing is size selective, and that means that you end up with a population in the water that is slightly smaller. And if there is a heritable component to this, and the smaller fish have a higher chance for reproduction, of course, what you can envision is an iterative process leading to selection for smaller size. Now, another body of evidence comes from laboratory experiments, and some of these were really neatly done. And a really fundamental of these was one done already 20 years ago, when they took silverside fish into the laboratory, and in just 6 generations of selective breeding, they managed to select lines that were selected for being larger, or large lines that were emulating more fisheries-induced evolution that would only grow to smaller size. And this has been a really influential result, because it shows that if you pick populations from the wild in a lab and put them through a breeding experiment, you can easily change the size of these fish through selective breeding. Moreover, they recently sequenced all these specimens, and they could compare them, of course, with the original founder population. And what you see here is a graph of the genomic responses of this breeding experiment, where we have 2 uplines, two down lines. On the y-axis, on the y-axis, we have a measure of genomic differentiation. On the x-axis, we have all the positions along the chromosome of these fish. And it's very clear that only in the control lines where this fish have been put through selective breeding, we see clear genomic responses that are absent from the control lines. And some of these regions are really, really large as well. Right, so where does that leave us? Well, we have both theory and selection experiments, which really suggest that it's possible for wild fish to be influencing rapid selection from standing levels of genetic variation. Moreover, it appears that these are really then targeting specific genomic regions, which suggest that we might have loci of large effect. Nevertheless, there are much more limited empirical wide capture examples. And this is, of course, where temporal genomics comes into play, because we really want to work with specimens that have been sort of present before all this event and before all this fishing, comparing the modern specimens to those old, to the historical material. So this is what we try to do. We have samples. These were kept in historical collections. We know really well where they're coming from. And I like to emphasize, this has been a really neat collaboration with your boss, Brendan Malin. It's been wonderful. We worked on this for years, and we're so happy that we finally got this out. So we looked at two different populations on either side of the Atlantic. Some samples were from 1940s in Canada. Some samples were from 1907 in the East Atlantic. And these are also two populations, I just showed you the graph before, that have experienced these rapid phenotypic changes. So these have often also been used as a key example of fisheries-induced evolution, at least from the phenotypic side of things. So we did a whole genome sequencing approach. The DNA of these specimens is really, really good. So it's really easy to get whole genome sequence data on a temporal perspective. We sort of have 8 to 10-fold coverage for each of these specimens. And we sort of span like 8 to 11 generations here. And I just actually added that because, Alison, I thought you had such a wonderful talk, and you can span so many more generations than we can here in the wild. But anyways, this is the reality of the system that we're working with. Okay, so yeah, this I think is that. So the first thing that we wanted to do, and even though we have really good metadata of these specimens, and we know we picked the specimens from the same location that the current population is, we really wanted to investigate whether we do have genetic continuity or population continuity, so to speak. So where do the historic specimens that we've sampled, are they truly closely related to the modern specimens that we've also obtained from that same location? And here you see sort of two clusters, Canada cluster and a Norway cluster, and you can hardly see the historical specimens, sort of the cluster underneath the modern specimens, which really suggests that at least we're sampling something that is very similar to the population that we can obtain from this ecosystem today. We can also do a simple admixture analysis, and this sort of shows the same thing. We only see a spatial divergence between Canada and Norway. Sorry, I also have to say that there is only one PCA that is actually significant. The other PCAs are absolutely non-significant, and they usually show rubble. So also from these two analyses, this really suggests that what we have here is something very similar to the population of today. And I think this is a prerequisite, because we want to know that what we're sampling now is something very similar to what we were sampling like decades ago, and that these populations have not spatially shifted over time as well. All right, so we also estimate fairly large estimates of effective population sizes. We use a temporal frequency comparison here, and for Norway, we estimate an average effective population size of around 10K. For Canada, it's even higher. We estimate 35K. And that, of course, indicates that genetic drift is not really going to be a major factor in this system. These are huge numbers, and these basically mean that we don't really observe a lot of genetic drift. We also don't observe any genome-wide loss of genetic variation. We can look at genetic variation, whether it's pi or Tejinos D, and we don't really see any major changes over time in either of these populations. So we then proceeded to look for selective sweeps on a more local level, and we used a range of different approaches. We used window-based analysis. We looked at FST, we looked at genetic diversity pi, we looked at increases in linkage disequilibrium, we looked at changes in Tejinos D. All these were window-based. We were also interested in shared outlier regions. Can we increase the power by using this parallel system? So are there some regions in the genome that are affected more, both on the western side of the Atlantic as well on the eastern side of the Atlantic? Then we also looked at individual levels. We looked at FST for individual SNPs, and we used different tests for that. And we also, of course, implemented a PCA-based test based on genotype likelihoods instead of hard genotype calls, which is implemented in ANGUS-D, also on individual SNPs. All right, so that then leads me to some of these results. And I'm only going to show you the FST differentiation. This is a Windows-based analysis, because most of these results all look identical to each other if they not really have they have slight differences between them. Here you see on the x-axis, you see the different linkage groups that we have in Atlantic cod. On the y-axis, you see a measure of genetic divergence. And the size of the bubble that you see is the number of SNPs in each of these windows. And we only sort of find 4 shared outlier regions, both in Canada and in the Eastern Atlantic, which suggests that these might be outlier regions, but there's only very few SNPs underneath them. And what's more, if we look at the overall picture and all our analysis, we have something of, I think, of a bit of a problem. And the problem is the following: there is no consistency between any of our outlier methods that point towards the same genomic region. Most of the outliers, if not all, these are singleton outliers. And this is a problem, because if you have a singleton outlier, you may have bias underlying such an outlier, and you're not really sure what goes on. Outliers are also poor in quality. They are poor in genome coverage. They are poorer in missingness. They are poorer in quality, missingness, and depth. And everything, of course, is associated with each other here. And we don't find any flanking regions associated with these specimens, with these regions as well. So there is no classical genetic sweep. And this is somewhat different from, for example, your rabbit paper, Joel, where you had multiple SNPs in the same region supporting outliers. And this, of course, is a much more classical sort of form of a selective sweep, which allows you to get the gain of trust of what you have, and that you don't have a risk of false positives, which I think is really something that we see here. So we interpret some of these outliers really cautiously, because I don't really know what goes on. And if these are truly outliers, and given the fact that they're significantly poorer quality, for me would suggest that these actually are false positives. And it's difficult to claim loci of false effect in this data set. All right, so what leaves us then here? Do we find direct evidence for fisheries-induced evolution over time using these temporally spaced data? Well, I think that's difficult to say. We can't say no. And the reason is our data set is a bit underpowered. And Alison, you've used your Fisher-Wright model to figure out what sort of the selective parameters were. We did the same. And we actually figured out that we have very little power to detect loci of very small effect. So this is, of course, a bit of a sad situation. We've spent a lot of time sequencing these things, and in the end, we may have to have sequenced thousands of more specimens to get at that loci, at those loci of small effect. But then it would suggest that genomic responses will not going to be super fast, because if it's a loci of small effect, it will take many, many generations for such loci to be fixed. And I think this is actually a key result, because it means that we're not going to lock our population into a specific genetic scenario whereby information is lost that allows fish to grow larger. And I think this is a positive result, because it would suggest that the phenotypic changes that we see, even if they are built upon by many losses or by many shifts in small frequencies of many different alleles, this is not then leading to an irreversible loss of genetic variation, at least not in this window of time. Of course, we're only looking at 11 or 8 generations over time. But in that sense, management change and actually not removing as much fish as we do in the current situation, I think would certainly lead to a reversal of the phenotypes that we have observed over the last decades. So, and with that, I'm actually coming towards the end of my talk. I don't even know how long I talked for, but I think it should be roughly 12, 15 minutes. I didn't check, but Brandon seems to be happy. So then I would really like to thank all the people involved in this project. This has been a really long project, and especially Malin Pinsky, who went through with me, and we've looked visually at many of these outliers and looked at the variation underlying them. We're like, well, we don't trust the region that this thing is mapping to. So then, so that's been what we've been doing for a long time together. So if you want to read more, there is a paper that came out several weeks ago, a few months now. There also was a really nice nice commentary by Jeffrey Hudkins and Anna Kuperinen. So if you want to read the paper, do read the commentary as well. It's really fun. And then I'd like to thank you for your attention, and we'll take talks after Evan. Sorry, questions after Evan.
Thank you, Bastiaan. Really, really interesting talk. And I think it's particularly interesting that even though you don't have like a clear result on that, I think your paper brings so much more questions than anything else. So that's why I think it's quite refreshing. I'll pass now the word to Evan. Evan is now a postdoc at the Center of Geogenetics in Copenhagen. He's in the Fernando Rassmann group, and he worked with a wide range of organisms from rabbits, pigs, dogs, and is now working with an absolutely giant data set of human data, both ancient and modern. So Evan, off you go.
Oh, thank you, Joel, for that nice introduction. And thank you to the seminar organizers for the invitation to be here today. So I'm going to be talking about something really at the kind of the opposite end of the spectrum in terms of the temporal scale that I'm looking at. And I'm working with human data here. And we've been trying to experiment with methods to resolve selection in a way that is aware of changes in ancestry through time, focusing in this case on the Holocene epoch in West Eurasia. So for those of you who are not like fully conversant with West Eurasian or European kind of prehistory, this period of time sees the end of the last glacial maximum and the retreat of glaciers in Europe, and the recolonization of this land by hunter-gatherer populations, and then the transition out of the Neolithic into the beginning of, out of the Mesolithic into the beginning of the Neolithic, where people adopted sedentary farming lifestyles and started living in close association with animals. And it's really hard to overstate how significant this period of time was in terms of the changes in human lifestyles. And it's kind of broadly speculated in the literature that these changes would have been come in parallel with substantial changes in the selection pressures for a whole range of different complex traits, and particularly exposure to newly emergent zoonotic diseases and things like that. So we have this kind of this study system where we expect the human genome to be under quite strong selection pressure, but it's not really clear to what extent that's true. And there's been a lot of advances in the last 10 years or so with genome-wide association studies that have identified really large numbers of variants within the human genome that are associated with traits of interest. But to what extent they're under selection, it remains an open question. So to kind of get at this question, we compiled a very large data set, well over 1,000 ancient genomes, some of which are novel to this study, sequenced by colleagues at the University of Copenhagen, and others are obtained from published studies. And for anyone who's worked with ancient DNA data, you'll know it's really tricky to do selection studies with this, because the data is really low quality, it's low coverage, it's gappy, and it has a high error rate. So working with collaborators at the University of Lausanne in work led by Barbara de Mota and Olivier Delanoux, they applied their newly published phasing and imputation method known as GLIMPS. And so this allows us to take our really poor quality ancient DNA samples and combine them with a very large modern reference panel and impute them to get high quality diploid phased haplotypes. Okay, so we've done that to our data set. We have this big data set with millions of high quality SNPs. And because we're interested in looking at selection for traits, we went to the human GWAS catalog, and this kind of confusing figure is a visualization of all the trait associations that have been found in humans, mapped against the chromosome. So I think what you can take away from this is that the human genome is really very highly polygenic, and that there are lots of SNPs that have very small effect sizes that contribute to modern traits. So we did some kind of variant curation on this and ended up with a list of about 37,000 independent SNPs that we modeled to reconstruct the allele frequency trajectories and the selection coefficients. So one of the really nice things about working with phased and imputed data is that you can use a class of methods that have become popular in the last couple of years, known as ancestral recombination graphs, or ARGs. And here we used a method published 2 years ago in Nature Genetics by Leo Spiedel and Simon Myers at the University of Oxford. It's a method known as Relate, and is a kind of, I won't go into too much detail about how this works, but the schematic along the bottom there gives you a sort of intuitive idea of as you move along a set of genomes, the topology that relates those samples to each other at each locus changes due to recombination. And so what you end up with is a series of these marginal trees that capture the history of mutation and recombination across these genomes. So we initially built an ARG based on the 1,000 genomes phase 3 data set to get a very high resolution estimate of the change in effective population size across the last 10,000 years or so in West Eurasia. And we used that to infer selection, initially based just on modern data, and then using a method known as clues, developed by Aaron Stern and Rasmus Nielsen at the University of California in Berkeley. And Aaron's done some really excellent work in the last year or so, extending clues to allow it to take as an additional input a series of ancient DNA, either genotype likelihoods, or in our case, genotype probabilities from the output of this imputation process. And so this allows you to both run clues either just on a series of time series data of genotypes, or on the ARG, or on a combination of the two. So this result I'm showing here, or this figure, is a Manhattan plot. In the top panel here, this is showing the negative log 10 p-values for the GWAS SNPs that we looked at. And in the bottom panel, we have these control SNPs. So for each GWAS SNP that we looked at, we paired that based on its derived allele frequency with a putatively neutral SNP chosen at random from within the chromosome, as long as it was 50 kilobases from the nearest gene region or GWAS SNP. And so what you can see here is that the GWAS SNPs are substantially enriched for selection over and above what we see in the control group. And here we find 14 genome-wide significant sweep loci, which are kind of broadly consistent with previous findings in this space. So nothing too striking here. But one of the things that we're thinking about, I think, is a problem with any kind of time series data is that you have an assumption there that your early sampling is ancestral to your later sampling. And if you have breaks in continuity of the ancestry there, or you have significant periods of admixture or migration, then that violates the assumptions of many of these models. And so working with collaborators at the University of Cambridge, in work led by Alice Pearson and Richard Durbin, Alice has developed this this kind of simplified demographic model of human prehistory in West Eurasia. And I'm not going to go into too much details about this, but we have these kind of 4 main ancestral populations that are highly differentiated from each other. These are the Western hunter-gatherers, Eastern hunter-gatherers, Caucasus hunter-gatherers, and Anatolian farmers. And these populations all existed sort of side by side, geographically spread out across West Eurasia, and then in subsequent periods of time, they admixed with each other in various fractions, and have all contributed differentially to present-day West Eurasian genomes. And so what we did was we took this model developed by Alice, and we used it to paint the chromosomes of our ancient samples. So build an ARG including our ancient samples, and then use a method that's broadly similar to genealogical nearest neighbors. So you have some labeled tips, so individuals you know come from an ancestral population, and then you look at who their nearest neighbors are within the ARG, and you can use that to infer the haplotypic background that each SNP is occurring on within these models. And we can then condition our selection analysis based on SNPs having these specific ancestral paintings. So this figure is very similar to the one I showed you before, but in this case, what we've done is we've conditioned the selection analysis on haplotypes having each of these 4 marginal ancestries, and then for each SNP, chosen the ancestry in which it is most significant, and then used that to make this figure. And so one of the things that one of the kind of takeaways from this is that when we do this conditional modeling based on the marginal ancestries, we find 23 genome-wide significant sweeps instead of the 14 that we saw in the pan-ancestry analysis. And so this really kind of indicates that the selection dynamics in the last 10,000 years or so in humans have masked evidence for selection at many of these loci. And that selection can be occurring in one of these marginal ancestries, but that can be washed out by social historical processes of admixture and migration between these populations. Okay, so I'm going to very briefly give 3 examples of this, where we're going to zoom in on some well-known loci in human genetics. So this is the MCM6 locus, which is upstream of the LCT gene, and the SNP at this locus is causal for lactase persistence. And this is maybe one of the most famous evidence of selection in humans, and nobody really has an adequate explanation for why lactase persistence would be under such strong selection as it is. And on the left-hand side here, in the plot labeled all, we see the pan-ancestry analysis, where there's this very strong signal of selection beginning kind of a little over 6,000 years ago and continuing up until the present day. But when we look at it in the marginal ancestries, we see that this signal is really driven primarily by a sweep in the Eastern hunter-gatherer background and in the Caucasus hunter-gatherer background, and that we don't see strong evidence for this particular sweep in the other marginal ancestries. But what's kind of interesting here is that the marginal ancestry that shows the strongest evidence of selection, this green variant here is not the SNP that's causal for lactase persistence. So what we think is going on here is we actually have multiple overlapping sweeps where there are potentially more than one target of selection occurring. So another example is the SLC45A2 locus, which is causal for light skin pigmentation in West Eurasia. And here we see quite a different story, where all of the marginal ancestries show strong evidence for selection at this locus. And so this suggests that the drive mutation occurred a very long time ago, before these populations separated from each other, and was segregating it at low frequency. And then there was a parallel sweep in these populations occurring across a very broad geographic range. And in the third example, we can have a look at the FADS2 locus, which is associated with lipid metabolism, and a lot of work done on this around the sort of the dietary transition to farming. And once again, here we see a really kind of complicated story where in the pan-ancestry analysis, we see this, this nice consistent sweep. But when we look at the marginal ancestries, we see that, really the strongest evidence of selection here is in the Eastern hunter-gatherer background and in the Anatolian farmer background, and that there's kind of weak evidence for selection in the Caucasus hunter-gatherers, where it was already segregating at levels that are quite similar to what we see in the present day, and really no evidence for selection at this locus in the Western hunter-gatherer background. And so there's kind of three broad takeaways, I guess, you can take from this work. And the first is that ancient DNA and time series data in general, really considerably improves our power to detect variants under selection. I didn't go into the details of the selection analysis we did just using modern data, but we didn't find anywhere near as many loci as when we looked at the time series data. The other takeaway is that admixture between ancestral populations has masked evidence for selection at many loci. And this kind of makes sense. You wouldn't expect signals to persist if there's been a wide-scale population replacement through time. and that multiple selected alleles may be common within genome-wide significant sweep loci. I mean, to get p-values that are really large enough to be found in a genome-wide test like this, you have to have extremely strong selection, at least extremely strong from a human genetics point of view. I mean, nowhere near the selection coefficients that that Alison was seeing in her data. I mean, like orders of magnitude smaller, but still very strong selection coefficients. And we think that actually enriches for loci that have multiple causal targets, and that those signals are the ones that we have the best power to detect. Okay, and with that, I'd like to thank the many people who contributed to this work as part of a large consortium project. And yes, thanks to the Rasimer group, the Center for Geogenetics for all of their support. Thank you.
Thank you, Evan, for such a great talk, and thank you the other two speakers. Really interesting talks. We already have one question on the chat, but so this is the time that, well, people can start asking things. I'll start with a question on the chat since it was the first one that was already put here, and I'll go through all the different participants. So yes, please, if you raise your hand if you have a question. So the question for Bastiaan start, could you, from Sabrina and Inta, could you please elaborate more on your conclusion? Phenotypic reversal constrained by reversible loss of genetic variation. Aren't there no reduction of genome-wide diversity based on Pinksy et al. 2021? And sorry if I missed something.
Yeah, no, sorry, I probably didn't explain this properly, but no, there were no genome-wide losses of genetic variation, not even non-neutral losses or selective losses. So we didn't find any. There was no significant loss in genetic variation, not that we observed. So, yeah.
Okay. Anyone else questions? If not, I can start already with Bastiaan since you're already unmuted. So I guess my question is about expectations, because I think we started from a very similar point of view in the projects. We both didn't really not so much what to expect. We have different, well, massive event of selection, slightly different in terms of coefficient. But my question is, what did you expect? Were you surprised with this result? And my second question is that I can see on your website that you're now exploring this question with ancient DNA. So what do you think the ancient DNA will add to this? And I imagine this is somehow even related with Evan's conclusions on some of his data.
Oh, good question. So what did I expect while doing this project? So this project went on for a long time. And one of the reasons was that, of course, we didn't get a strong signal. And then you have to start wheeling through. So what is it that we actually see? And I think that taught us a lot. But on the one, when I started this, I thought we're not going to find anything. But on the other hand, then you saw all those major effect loci in the experiment. Like there must be something. But if you start thinking about these populations and how huge they are in compared to terrestrial systems, and if you also look, for example, at patterns of linkage disequilibrium, these are highly outbred individuals. And they have, even if they have removed like 90%, sometimes 95% of those of the biomass of these species, there are still thousands and thousands and thousands of them left. So you can change entire ecosystems, I think, in the marine environment by removal by physical removal of these fish, and still you have fairly large numbers that might still be reproducing. And I think therefore, changes in frequencies and equilibrium drift frequencies, they take a very long time to reach a new equilibrium. And before you pick that up, this is really, I think we need more generations before we would see such things. Yeah. As in terms of other ancient DNA, yeah, I'm really looking into this using other approaches, but of course there we go also into an area where we then may have longer generational time, but we're sitting with much more complex data. Like Evan is, you're sitting maybe with a question like, are these actually the ancestral fish of the population that you currently sample? Lots of more questions, and we need to dig into that. What I'm really interested in there is also use low coverage data to also incorporate a more integrative approach with archaeology, because I think one of the things that I'm really interested in is this concept of ecological globalization. When people started moving fish around, when people started doing trade, it means that you unlock sort of you unlock the you go from a sustenance hunting that is always limited because it's local to a situation where you can remove very large amounts of biomass because it's very beneficial for you. And this, of course, changes the way that we deal with ecosystems. And I think we're now sitting on a pattern where we have hundreds of samples all over Europe that we're going to trace back and figure out where were they from, what populations were they from, and we can do this at an increased detailed rate. Yeah.
Thank you, Bastiaan. Evan, you have a question, I think.
Yeah, I just had a quick question for Bastiaan, and maybe this is sort of coming from the sort of human GWAS world, but like if we can imagine a scenario in which you could go out and collect a very large number of cod and do a GWAS on body size, do you think you'd see something similar to what we see in humans, where, for instance, height is a highly polygenic trait with many hundreds of independent loci? And if we saw something similar in cod, then I guess maybe what's going on there is polygenic adaptation, where you just have these these very small effect loci that don't have to shift much in their size. And do you think that's what's going on here with these fish?
Possibly. What I think is really difficult here is that what we have in the lab, where we saw these loci of large effects going through, or major effects on the genomes, is that we can't emulate, I think, what we have in nature. So what you have there is many outbred populations, and we also have, for example, it would be, well, for example, size itself is a very difficult trait in fish because they keep growing. So you would also have to use some sort of a comparison on age. So you have to make sure that you got your cohort right. It's very phenotypic, very plastic. So in terms of and then I think also what you do when fishing, you're fishing on the adult individuals at the end, sort of, at their lifetime. But in order to reach that, there is tremendous pressure for fish to grow large, because the larger you are, the less risk you have to be eaten by something even bigger. So I think that whole sort of the selection during the lifetime of a fish I think it's very difficult to emulate in the lab. And I think there is also opposed sort of selection pressures that we might put in on the adult stage, but that is counterproductive at the younger stage. So in that sense, how much selection pressure really there is over a lifetime of a fish, I think that's very difficult to really assess. Yeah.
Katie, I think you have a question.
I had a question for Alison. I really liked how you used ABC to ask the question, from this empirical data, what could migration and selection be? But I was just trying to understand, when you were monitoring migration, it was between tissues within an individual, is that right? And so there's still the step to look at also the migration between individuals in the population, correct? So how are you thinking about that?
Yeah, thanks for the question. So the migration between individuals is, I think, maybe in analog to what we're seeing with SARS-CoV-2 evolution, has sort of a different process in terms of the things that make you successful within a host may not necessarily make you successful migrating between hosts. And one of the things that we see in sort of a global phenomenon in HIV is that things that are transmitted between hosts tend to be closer to the global ancestry, like the global consensus strain, as opposed to things that are really acquiring a bunch of intra-host mutations, just because the HIV mutation rate is ridiculously high. So, I think that there's definitely a lot of work on understanding, for example, transmitted drug resistance, which is becoming increasingly common as antiretroviral therapies become increasingly widespread. You're right. You're totally correct, though, to pick up on this fact that understanding the sort of levels of where migration is taking place is a really crucial factor. And one thing that I'm particularly worried about is actually migration within even much smaller scales. Like, what does it mean to migrate from one part of a lymph node to a different part of a lymph node? Like, how are we able to account for migration, not just between these easily definable places, but these things that are a lot more challenging. But yeah, I don't have specific plans on between host migration, but it's definitely super important in understanding global patterns of drug resistance.
Thank you. Bastiaan.
Yeah, I had a question for Evan, because I'm always so jealous on the way that you can do stuff with human data that is more difficult to do what you can do with species that are not so well known. So one of these things is imputation. And I was wondering two things. What is your error rate, you think, in imputation? And is there an error rate associated with imputation? And two, imagine there is variation that is lost. how can you impute genetic variation back in time that may not be part of your reference panel that you used for imputation?
Yeah, no, those are two good questions. So in terms of the error rate, there are methods that we can use to estimate that empirically. So I mean, one of the most straightforward ways to take a high coverage ancient sample and to downsample it, and then to feed it to the imputation algorithm and measure the correlation between the true genotypes and the imputed ones. And under those kind of simulation scenarios, the imputation quality is surprisingly good, at least for ancient individuals that are kind of well characterized in the modern reference panel. So I think, imputing West Eurasians from the last 10,000 years, you can go down to as low as 0.1x and still get pretty reliable coverage, as long as you do fairly aggressive filtering on the SNPs afterwards. The accuracy is really dependent to a large extent on the minor allele frequency of the SNP within the modern reference panel, and how close those haplotypes are to each other. In terms of your second question, there's a lot of ascertainment bias that's present in work like this, because it's not simply variation that might have been lost in the past that we know nothing about, but that's also a problem for our GWAS, because we can't identify trait associations that for sites that are no longer segregating in the present. And for a lot of human complex traits, the modeling and the theory suggests that these are under a mild negative selection, particularly for something like height. So the selection is purging slowly large effect alleles, and you get really many very small effect alleles. And so it's kind of doubly problematic there, because we're probably underestimating in present day GWAS the number of causal variants, because they've been lost or because they were at high frequency in the past due to this process of negative selection. So there's a lot of problems with that, and I think there's a desire within the ancient genetic community to kind of move towards using polygenic risk scores to actually kind of quantify complex traits in the past. And I have a preprint up on bioRxiv at the moment where we review this kind of work in the literature at the moment. And I think it's really problematic. I think the known unknowns in this work are very large, and the unknown unknowns are potentially even more confounding. And as much as we may want to make these predictions about the past, I think we need to be honest with ourselves and with the broader research community that that these predictions at the moment are really heuristic and noisy, and shouldn't be taken too seriously.
Thank you, Evan. Actually, I have also a question for Alison. I was wondering if there are any mutations in HIV or any kind of virus that can affect their ability to proliferate or to survive better in different types of tissues. And if that is the case, how might that affect some of the dynamics that you observe?
Yeah, good question. That's something that we actually set out to try to understand. So I'm not sure if you were looking at the sort of the timestamps of when these different work was coming out. We first had this sort of qualitative picture of how much or what the description of each of these viruses in these different tissues was first. And one of the big questions was, can we produce this using, do you need tissue specific adaptation in order to create these dynamics? And so the point of the follow-up project to actually do the ABC was to understand whether you did need to invoke tissue specific selection in order to create these effects. And the answer was you don't, if you have really rapid evolutionary processes that are happening sort of in parallel in a bunch of different places that can create these effects that sort of cause FST to go up and then go down over time. But I think that there are good reasons to believe that there might be different effects of different mutations in different tissues. So for example, one of the hypotheses we have about how HIV evolves multi-drug resistance is that there's actually different penetrance of different drugs to different parts of the body. And notably, you might have less penetrance to areas like the lymph nodes than other places. And if that's the case, I'm not sure you would necessarily expect like a sign reversal of drug resistance mutations in different places, but you would expect the selection coefficients to be different. So if, for example, in one area of the body, beneficial mutation provides a huge selective advantage because the drug is really prevalent versus if it only provides a very weak selective presence because it's not getting there to a high degree of efficacy. So that's a hypothesis we're really actively working on. But what we found in the SHIV data, at least, is that you don't need to invoke any of that in order to explain those patterns.
Okay, excellent. Thank you. Are there any other questions? If not, I'll ask at least one to Evan. So I'll make sure I ask a question to all the different speakers. And it's a very convenient question to ask Evan. And you're hostage of this talk now, so you can give me your advice. So from the perspective, since you're dealing with this type of data, you have a massive amount of data and you have some of the best data people can use on ancient DNA. If you were going to sort of try to address this question of selection over time, how would you, and you have access to a lot of archaeological samples across different time points, would you go for large populations on a specific time point, or would you go to rather have more individuals at more different time points? Which of this would be more robust to actually try to track selection over time? And I understand it's a difficult question.
Yeah, I mean, I guess I'm going to give you an inadequate answer to that, or an unsatisfactory one. That it really depends on the nature of the question that you're asking, and you know, certainly in a very long temporal transect, you have more power to detect you know, large changes in allele frequencies, but that may not be, you know, what's biologically occurring within your study system. So it really depends on what your hypothesis is about the broader environmental effect that's driving selection. You know, you definitely can identify selection in shorter time periods, as you've shown yourself, Joel, in your work on the rabbit evolutionary response to myxomatosis. But yeah, I think, I mean, in terms of what you have statistical power to detect, yeah, deeper transects give you more power, but only if the size or the magnitude of the change is large.
Okay. Well, thank you. Is there any other questions?
I had, first of all, I just wanted to say great talks, everyone. I just had one question for Alison, and and it's interesting when I usually think about like soft sweeps. So I'm thinking about organisms that have much slower generation times and much smaller population sizes, where kind of the allele that's under selection has to be kind of there already. And I think you were kind of talking about it in a slightly different way, where it's just the waiting time is so short because there are so many and you know, they mutate so quickly that you can have those multiple different those alleles just kind of arising and then sweeping and then, you know, changing so rapidly. So I was wondering too, though, can you differentiate those sweeps where it's already present at some very low frequency in the population from the sweeps where it's an actual new mutation and it's just arising very quickly? And how would you do that in in a virus where you just have so many? How do you sequence enough?
Yeah, good question. So there are sort of two things going on here. One of them is that we know that these mutations have huge fitness costs in the absence of the drug. So we expect that they're not at super high frequencies in the absence of the drug. That being said, there are some drug resistance mutations that can get transmitted, and it's not a totally absent phenomenon, and that it is possible. There's a cool study that one of my co-authors, Pliny Pennings, did, I think about almost 10 years ago now, in which he's asking exactly that question about how do you distinguish standing genetic variation from de novo mutation. And one of the ways that she approached it is that she looked at patients who were failing therapy in their first, second, and third year of treatment. And she found that things were extremely consistent between the second and third years of treatment in terms of the percentage of patients that acquired a new drug resistance mutation. However, there was an excess of failures in the first year of treatment. suggesting that at least some of the failures that occur in patients in those are likely from standing genetic variation, whereas in the patients who had been on therapy for a full year, it's unlikely that standing genetic variation was the explanatory factor. So if you can subtract out basically the rate of failure in the second and third year from the first year, you get a proportion that they think is from standing genetic variation, and that's how they approach it. But yeah, in general, it's a challenging thing, in part because there's so many mutations on so many backgrounds.
Thank you.
Is there any other questions? Well,
I had another question for Alison, just a quick one. When you're talking about migration, I guess it's a different sort of migration to what I kind of think about in traditional population genetic view. And so, if I understood correctly, you're talking about actually like physical movement of the virus within the host. So how do you distinguish that from independent de novo mutation occurring in separate parts of the host?
Yeah, I think that it's definitely possible that independent variants are emerging in different parts of the host, and it's hard to tell those apart, especially if they have exactly the same genetic background as in the case for example, that M184V mutation in the case of the SHIV. If it's exactly the same mutation and it's on a totally non-diverse background, then you can't really tell it apart. Luckily, if you have whole genome sequences, you generally do have passenger mutations that allow you to do a little bit better. In the simulations that we were comparing these things to, we were mostly just looking at measures of FST, at measures of the amount of sharing of distinct haplotypes, which can be created via identical mutations in different backgrounds where it looks like there's been a migration event, but really it's a recurrent mutation. And the hope, I guess, is just there that the ways that we were doing this by having a barrage of summary statistics that are capturing different aspects gives you some resolution there. But I agree that it's a hard problem, and that there are definitely situations in which recurrent mutation can mimic migration exactly.
Any more questions? Well, if not, before I pass the word to Brendan, I'll just really thank you, the three of you, Bastiaan, Alison, and Evan, for such amazing talks and such fascinating topics, and everyone that asked questions. I think it was a really productive and interesting discussions, and it's one of those that we get out of here, at least me, with more questions than which is a good thing, I guess. So, Brendan, do you wanna wrap it up?
Nothing much else from me except thank you again to everyone, and yeah, thank you for showing up, and thank you for all the great work, and I really am interested in where all of these projects are going in the future. So thanks, guys.
Yeah.
Thanks,
guys. Have a great weekend.
Yeah.
Bye.